Chapter 19, Oxidative Phosphorylation. Now, when we look at this idea of oxidative phosphorylation, it is thought of, considered, taught with the idea of the chemiosmotic theory as the centerpiece for how we think of it, how we discuss it, how we study it. The chemiosmotic theory holds true, to the best of our knowledge, to all eukaryotic organisms. You look here in this figure, it's supposed to represent, a mitochondria. So eukaryotic organisms, this is what's going to happen in the mitochondria. We also have to keep in mind that prokaryotes are going to have their own version of this. And so instead of this occurring at the mitochondria, since they don't have organelles, the prokaryotes will be happening at their cytoplasmic membrane. This chemiosmotic theory is based on the idea that you're going to have concentration difference across a lipid bilayer barrier. High concentrations on one side, low concentration on the other. Here, it just happens to be protons / hydrogens. High concentration hydrogens on one side, low concentrations on the other. Positive-ish on one side, negative-ish on the other. So, positive, negative. This sets up an electrochemical gradient that is separated by a lipid bilayer. As I said, this lipid bilayer here could be the mitochondrial membrane, inner membrane, the cristae, the inner membrane, or it could be the cytoplasmic membrane for prokaryotes. To set up this gradient, this difference, you're going to have a series of proteins forming these complexes that are going to be pumping hydrogen. Remember, all state of matter flows or diffuses from high to low concentrations. That's just the natural state of matter in this universe. To go against that gradient is like pushing a ball up a hill. Takes work. So to push means you're going to have to pump hydrogens from low to high. To do that, you're going to need energy. Where's that energy coming from? A high energy electron. That high energy electron has been oxidized, stripped off of a sugar, amino acid, an acetyl-CoA from a fatty acid, or this or that or something. Why am I being so vague? Well, because it's going to be different. Cell type to cell type, prokaryote to eukaryote. That's what the past 5 chapters have all been discussing. So high energy electron is going to be passed from one protein complex to another. As it's being passed, a little bit of energy is going to be bled off. That little bit of energy is going to be used to pump hydrogens from low to high. You have this differential, extremely high, extremely low, and things are going to diffuse. Well, hydrogens are slow to diffuse through the membrane, but they're very quick to move through ATP synthase. As we're going to see in this chapter, the movement of the hydrogens from high to low, from extremely high to extremely low through ATP synthase is what's going to give the energy required to take a diphosphate, inorganic phosphate, put them together to make ATP. So let's start talking about that respiratory chain, also referred to as electron transport chain. You'll hear me go back and forth. More often than that, it's always been taught to me, discussed with me, and I've always discussed it, talked about it, lectured about it as the electron transport chain. But as we've seen in previous chapters this semester, last semester, electron transport chains can occur in many different places. Here, oxidative phosphorylation, you also see them, nitrogen reduction, you also see this in photosynthesis, the light reactions and whatnot. So just keep in mind for this chapter, I may slip in and it may slip out as electron transport chain. What I'm referring to is the respiratory chain. Everything's occurring here at the mitochondria. Remember, hopefully you remember from your endosomal cell biology, that the mitochondria is a dual membrane organelle. Lipid bilayer, another lipid bilayer. The internal lipid bilayer is also referred to as the inner membrane, is actually much, much larger than the outer. Since it's much, much larger, that means there's lots more surface area. And to get that much larger inner membrane to fit, it has to be just shoved in here. And you get all these folds that are referred to as the cristae. What's so important about these cristae? Well, as I just said, more surface area. More surface area means more real estate. Location, location, location. This is where all the protein complexes we're going to be talking about in this chapter will be parked. Also, you got to remember, you have this gap between the outer and the inner. So everything we're talking about is occurring right here in these cristae, on these cristae, through these cristae, and things are going to be taken from the inner space here, known as the matrix, and then moved to this intermembrane space. Nothing we're going to be talking about, I say that nothing, take that with a grain of salt. Nothing we're talking about really is going to be going out in here into the cytoplasm. It's into intermembrane space, back into the matrix, back to the intermembrane space, back and forth. The matrix of the mitochondria has quite a few enzyme and protein complexes that we've talked about before. Pyruvate dehydrogenase complex. Takes the pyruvate that's coming in, we'll go through and strip off an electron as it's going from pyruvate, as the pyruvate's being transformed into acetyl-CoA. All the enzymes of the citric acid cycle that make all those intermediates in the mitochondria. Fatty acid beta oxidation, we talked about in the previous couple of chapters. All of that occurs in the matrix of the mitochondria. Even the amino acid oxidation, matrix of the mitochondria. This is why these three cycles here I have been harping on for basically this entire semester. That all these different metabolic pathways, all these different cycles, how they can feed into and feed out of each other. And the reason they can and do it so well, they can and do, and while doing it, do it so well, is the fact that they are all in the same place. They are operating right next to each other. So if you have have a molecule, some carbon-based molecule, some organic molecule that's an intermediate in the citric acid cycle, but it's also an intermediate in beta oxidation, well, it turns out it's which enzyme gets to it first sometimes, because they're right there next to each other. And those cristae, okay? It's not just, oh, look, the artist drew them in. They're not really that big, not really that extensive. No, look, right here. This tissue scan where they went through with a computer and did different, colorations to heighten the differences. And you can see right here, all of those lines are cristae. Now, while the lines look like they don't go anywhere or they aren't attached to anything, they are projections inwards of the inner membrane. So they are all one continuous membrane. One of the things that helps, or actually supports, our theory of our endosymbiotic theory, where the mitochondria and the chloroplast come from, is the fact that the mitochondria actually controls its own metabolism, controls its own growth. So while the cell, the eukaryotic cell around it, is doing whatever that cell does, whether it's a protist, a fungi, a neuron in your head, doesn't matter. It's doing its job. The mitochondria has its own genome, has its own metabolism, and controls its own growth and division. Turns out that there's a cell type that's going to need lots of ATP, more than the quote-unquote norm. Well, that's mitochondria is going to go through and divide, make extra copies to keep up with the ATP requirement. Any of the mitochondria get damaged because, well, all cells get damaged by what you eat, drink, some injury to the organism, whatnot. Well, they will divide, replace anything that gets damaged, gets broken down. All of the mitochondrial components, the amino acids, the nucleotides, the lipids, because remember it has its own genome, it's going to have nucleotides. It has its own membranes, so there's the lipids. All of its own proteins, enzymes, cytoskeletal components, there's your amino acids. Now that respiratory chain, what I'll sometimes refer to as an electron transport chain, it's basically just in a series of carriers, electron carriers. And we've talked about these carriers, these little cofactors, these little coenzymes and things in previous chapters, because their job is to take something, hold it, and move it from point A to point B without it getting lost. Some of them are also going to be inserted into active sites of much larger enzymes. That way, an intermediate or a functional group that gets pulled off of A is still there and ready to be added to B. We see that for the dehydrogenases that are used throughout glycolysis, citric acid cycle, and all of the other metabolic pathways that we've talked about previously, that there are a set of these carriers that predominate. Nicotine amide nucleotides, commonly referred to as NAD or NADP, if NAD has a phosphate group or an inorganic phosphate group attached to it. It's the only difference between NAD and NADP. We also have the flavin nucleotides, FMN, as well as FAD. These nicotine amide nucleotide linked dehydrogenases, what they do is they catalyze the removal of an electron / hydrogen from something, and well, they got to do something with that hydrogen. They're not just going to release it, because by releasing free hydrogens, you're going to acidify that environment. And that hydrogen has a high energy electron. So what they're going to do is they are going to oxidize or reduce NAD to NADH or NADP to NADPH. We'll see that 2 hydrogen atoms will be removed. One is given to NAD, the other is released. So there will be some slight acidification, not much, but definitely nowhere near as much as if you're going through cleaving to and just releasing to. When you go through and you look, we've seen these, we've talked about these. You know, alpha ketoglutarate to succinyl-CoA. NAD to NADH in the mitochondria, mitochondrial cytoplasm, alate to oxaloacetate. In the cytoplasm, G3P to 1,3-bisphosphoglycerate. Cytoplasm. Mitochondria. They are a very major component. Now the flavoproteins, the FMN, FAD, they do roughly the same thing. Except their job, they take both the electrons that will be pulled off. FAD, H2, FMN, H2. The transfer occurs because remember, all of these are irreversible, left to right, right to left. Well, when you're having the reduction of FAD, because 2 electrons are being stripped off or oxidized off of, or a compound's being oxidized, it's losing the electrons and it's going left to right. more often than it's going right to left is because it turns out the flavoproteins have a higher E0 value. That means they have a greater pull towards those electrons than the initial substrate does. So yes, while that reaction can go left to right, right to left if you were drawing this out as a chemical equation, we see that it's more often than not going to left to right and staying there because FMN has the higher E0 value. Remember, this E0 value is a measure of which direction, who's got the greater affinity for that electron, thus left, right, right to left, which way is that reaction tilting. Okay, doesn't mean it can't go right to left, it just means it's tilted more to make it more favorable for left to right. So these carriers that are going to be part of this electron transport chain, this respiratory chain, you're going to see direct transfer of electrons from protein to protein to protein, complex to complex to complex. They're not going to be released at any point in time until everything is done. So we're going to see the transfer of hydrogens, we're going to see the transfer of hydride ions. Excuse me. Direct transfers. No, protein A takes it, does its job, releases it, protein B comes along and finds the hydrogen, binds to it, the electron binds to it, does something to it, and passes it on, releases it for the next. No, it is direct transfer. So, the electron carrying molecules that we're going to see involved here, NAD and the flavoproteins, FAD, FMN, we just talked about, ubiquinone, commonly referred to as coenzyme Q, or sometimes just Q, especially if you're reading a textbook or a primary literature paper that all they are talking about is a respiratory chain or electron transport chain. So instead of writing coenzyme Q, they'll sometimes refer to it as just CoQ, like CoA, CoQ, or they'll just get really, really lazy and just call it Q. Okay? Tomato, it's all the same. You're going to see the cytochromes are also going to come into play, and we'll talk more about this in this section of the chapter. The cytochromes are going to be a series of organic molecules. They're going to have a metal at the center that will accept the electron, will hold the electron and allow it to be easily released. And then you're going to see some iron sulfur proteins that are doing the same thing as the cytochromes, but here they're passing it along, aiding in the movement within the proteins and stuff that make up these complexes. So let's talk about the first one we haven't seen or discussed before. Ubiquinone. Okay, ubiquinone, coenzyme Q, CoQ, whatever. Same thing. It's a lipid-soluble benzoquinone with a long isopronid chain. Basically what that means, that nice little sentence right there means, is you have a hydrocarbon, as you can see here by all the hydrogen carbons and hydrogens throughout here. So this is extremely hydrophobic. And then you have up here a little benzene ring. It's here at this benzene ring that you have what they call the ubiquinone, the Q, okay? Because here's your carbon chain, and it's 10 of these repeated over and over and over. So it's quite a long hydrocarbon with the benzo ring at the end. The benzene ring here, carbonyl, carbonyl, carboxyl, carboxyl, methyl. It's the carbonyls, 1, 2, that will be modified, as we see here, to become hydroxyls. The incoming hydrogens / electrons, those are the excited hydrogens, excited electrons that are being delivered by NAD or FADH2. Ubiquinone, 1 carbonyl has been turned into hydroxyl. UB semiquinone, also referred to as QH. And then ubiquinol, QH2. Fully oxidized, fully reduced, semi-reduced. Now, since this is extremely hydrophobic right here, that means the best place to park this bad boy is in the most hydrophobic place in a cell, a membrane. What's our membrane of choice here to be working with? The inner membrane, the cristae. So this is actually going to be inserted into, moving throughout the inner membrane part of the lipid bilayer that makes up the inner membrane. So it can diffuse through, it can move through where the fatty acid tails are of a lipid bilayer. The cytochromes, as I mentioned a few minutes ago, we've seen similar to, okay? They'll have an iron-containing heme that will accept the electron, okay? This prosthetic group for the cytochromes we have seen before. It's very similar to chlorophylls that were discussed last semester. For those of you that are just taking it this semester, you haven't had 421 yet. Hopefully you remember your photosynthesis and the chlorophyll molecules from previous courses. What we're looking at here is different forms of cytochromes based on the structure of the ring. Now notice the rings. This is a large organic structure. Have the nitrogens, functional groups, pointing in here. And due to the orientation of these nitrogen groups, what they are holding here through electrostatic interactions is the iron. And it's going to be the change from ferredoxin to ferrous, ferric to ferrous, addition, subtraction of an electron that's going to change the state of the iron, allow that electron to be moved from one complex of proteins to the next without being lost. Cytochromes B, A, C all have this central ring structure in commonality, but it's what's attached or what's holding this ring structure here. It's showing, cysteine amino acids. Here it's different. Here it's a long lipid chain. And when we look at the absorption spectra, alpha, beta, gamma, ABC, and you can tell the difference between oxidized and reduced. No electrons carrying electrons. The iron-sulfur proteins, there are proteins that, due to their tertiary quaternary structure, they'll have a site that has cysteines that are all oriented here in such a way that they can literally covalently bond to iron. So it's not an electrostatic, as we saw with the cytochromes here, holding one iron in place. Here, the irons are held in place, yes, through electrostatic, but also some covalent bonds. And the structures get more and more complex depending upon how many irons are going to be held by these four cysteines. 1, 2, 4, knowing that each of these, 1, 2, 4, are going to interact with at least one electron themselves. Now, it was a long, drawn-out process. What, I'm going to be busting through rather quickly, probably, in this section, in this lecture. Here's the complex of proteins, and here's the sequence that, the different carriers are working, and all of this stuff. And unfortunately, it makes it sound like it was just, well, duh, here's how it works. We know this, move on. And it turns out it was a lot of hard work by people who are way smarter than I am. all the different proteins and enzymes and things you have in your cells, the majority of them are going to have a compound that can interfere with their activity. Somebody figured out that there was a set of these that will interfere with mitochondria activity, specifically when it came to oxidative phosphorylation. With those set of that interfere with oxidative phosphorylation, they figured out the ones that interfere directly with the electron transport chain. Well, as we've seen a couple of slides ago, different cytochromes have a different absorption spectrum based on whether or not they're oxidized or reduced. Well, it turns out the majority of these, carriers also have a different absorption or have different chemical signatures based on whether or not they're oxidized or reduced. So what do? They bust open cells, use differential centrifugation, isolate a bunch of the mitochondria, still intact, and then they went through and they put those mitochondria in different solutions containing the different inhibitors, rotenone, antimyosin, cyanide, whatnot. And then they let the mitochondria, because the mitochondria are still functioning. I mean, other than, taking up organic molecules from the cytoplasm, the mitochondria pretty much do their own thing and just pump out excess ATP. So the isolated mitochondria still went and did their jobs, just kept going and going and going as long as they were kept in a buffered system and were supplied with what they needed, oxygen, organic molecules, acetyl-CoA, things like that, so they can keep going. In the presence of these different inhibitors, what would happen is a certain step or a certain protein or here carrier would be interfered with. They can then go bust open those mitochondria, isolate all the different compounds, and look at their different absorption spectra to see which ones, if we added this inhibitor, which ones were reduced, which ones were oxidized, or in a reduced state, which ones are in oxidized state. And what they were able to do is go through and figure out, in the presence of this inhibitor, these two would be reduced, or in the presence of this, these ones would be reduced. These ones would stay oxidized, back and forth, be oxidized. These would be reduced. These wouldn't change. It's kind of like that question that was always on the ACTs, the SATs. Bob sat next to Jill. Jill has to sit 3 away from Tom. Tom has to sit right next to Dana. Dana has to blah, 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 blah. What order were they seating in? Well, that's what this was. Well, they were able to figure out NADH to Q, to cytochrome B, cytochrome C1, cytochrome C, A, cytochrome A3, and then the final electron acceptor, oxygen. What they found is the electrons tend to flow lower E prime naught to higher E prime naught, because remember, the lower the number for this voltage, the greater the potential. So NAD to NAD, NAD to NADH, NADP to NADPH, -3. Here, oxygen plus hydrogen plus electrons to form water, 0.8. -0.3 to +0.8. That is a really big difference. So you go through and you follow ubiquitone, NAD to ubiquitone to cytochrome B, C1, C, A, A3 to here, just the series, the sequence that we talked about last. You go through quite a change in voltage. So the order. NADH delivers, this is coming from the cytoplasm or from the citric acid cycle, doesn't matter, to Q, Q to cytochrome B, B, C1 to C to A3 to oxygen. And they've confirmed this over the decades. They have looked at this and they've tried different ways to study this, to inhibition studies, follow the electron studies, all of it.